Hello, my name is Zander Ribao, and I am a biochemistry and molecular biology and biological data scientist double major in the Colleges of Science and College of Agricultural Sciences. Today, I will be sharing my work on the biocuration of rice genes and pathways involved in salinity stress response for the plant reactive knowledge base, conducted under the guidance of Dr. Sushma Naithani. Now, how exactly does salt stress affect rice? While high salt stress can outright kill the plant due to severe osmotic and ionic imbalances, as well as reactive oxygen species, moderate and low salt stress can still affect the plant. At the vegetative stage, leaf burning or browning, as well as general damage to tissue, occurs, while in the reproductive stage, spikelet sterility and reduced florets and grain weight, all of which negatively impact crop yield. And when you consider that not only different species, but also different cultivars of the same species can have differing reactions to salt stress, consolidating the knowledge of the different salinity pathways that occur within rice plants becomes crucial for breeding a resilient staple crop that can continue to feed billions of people worldwide. With that in mind, I began working on my project, which involved me using a biocuration approach to synthesize knowledge about genes and pathways involved in salinity tolerance and susceptibility. Now, of course, you might be wondering, what is biocuration? Well, you can think of it as an iterative process that involves collecting, organizing, annotating, and validating biological data to make it accessible and useful for a variety of professional and applications, with biocurators leading the charge in interpreting complex experimental data and translating it into standardized, structured formats within biological databases. There are multiple steps involved in the biocuration process, ranging from data extraction, where the actual biological information from sources like scientific literature and the like are collected, all the way to the data's integration into centralized biological databases. My project can be organized into four steps, with the first being the critical review of literature to extract a list of genes as well as their overarching mechanisms. However, this is often not enough information, requiring the second step of annotation enrichment, in which various databases are used to embellish relevant information on the curated genes. For instance, GraphDB was used to identify the gene IDs, UniProt for the protein products, and CropPal for the subcellular location of these proteins. Other tools like BLAST and the Grameen Genome Browser were used to confirm the identity of genes if unclear or overlapping gene names were used in the literature, to ensure accurate curation and help homogenize the gene nomenclature to streamline eventual integration. Then, the genes and the functions of their protein products were used to draft their connections, where, in my notebook, connected their up and down regulatory patterns and interactions before synthesizing the information into comprehensive gene networks. This gene network that I made, and which you can see right now, features calcium signaling in rice and a plethora of other reactions occurring across the cells of rice plants. As it is hopefully apparent, the salinity response is very complex. Luckily, the plant reactive knowledge base can act as a scaffold for this information. The home page of the plant reactive knowledge base can be accessed by typing in the link on the screen into your address bar. It features the gene pathways of 138 other plant species based on gene orthology projections and is designed to be accessible to researchers around the world. By clicking on Pathway Browser, you will get a view of the interactive interface of all of the currently curated genes and pathways for Oryza sativa. To view the curated salinity pathways, click and expand the Abiotic Stimuli tab for navigating to the Response to Salinity tab. From there, you can explore the current curated salinity pathways. This is a view of the currently curated transcription factor network involved in the salinity stress response. Reflecting on this project that I worked on for 15 weeks, I was able to accrue experience in various genomic, proteomic, and transcriptomic databases, as well as learn how to critically review scientific literature. This research experience also helped me emphasize to how to think at the systems level and reinforce content relevant to my majors, like functional genomics and gene regulation. This all culminated with my poster presentation at the 2025 Spring Poster Symposium, where, besides preparing a scientific poster and presenting my research for the first time, I was also able to witness the diversity of scientific research occurring within the OSU community. For the future goals of this project, the information previously discussed will, over the coming weeks, be used to update the plant reactive knowledge base. And this current summer, I'll be conducting promoter analysis of these salinity response genes, as well as begin the biocuration of genes involved in rice's response to heat stress. Finally, I would like to thank Dr. Sushma Naitani for serving as my mentor, as well as URSA Engage at Oregon State University, whose funding supported my work on this project. Thank you.